In today's video, we're going to look at the structure of carbohydrates, and we're going to go through these objectives. So in terms of the lower sixth that I teach, this kind of vaguely follows the structure of the notes termed carbohydrate biochemistry. So it's one of the first lessons in your A-level course, and it follows on from water quite nicely for the reasons you'll see in a minute. So this may look like a lot of objectives, and it is, I guess, but hopefully some of these we can tick off fairly quickly as a result of you already covering the biochemistry of water. So we'll make a start. So the first thing we're going to look at is the classification of carbohydrates and the structure of glucose, which is our single carbohydrate, so something called a monosaccharide, which we'll look at now. So the first thing to do is do a little bit of introduction on glucose. So the first thing that I'm going to say about this is hopefully you're aware that this has the formula C6H12O6. There are 6 carbons, 12 hydrogens, and 6 oxygens. And you may have seen at GCSE that it is represented typically by a hexagon. And that is to reflect the fact that there are 6 carbons. And in skeletal formulae for chemistry, wherever there is a bend in the road, we infer that there is a carbon, unless we're told otherwise, at that position, and that there are hydrogens attached. Because this exists on its own, it's kind of its own structure here, as shown by the hexagon, we call this a simple carbohydrate. And the posh word for this, meaning that it sort of exists as a monomer on its own, is something known as a monosaccharide. Monosaccharide. And hopefully this is fairly straightforward, that mono here refers to there being one, in this case one unit, one sugar. And saccharide literally means, or it comes from the Greek, if I change colour, there we go, bit of an odd colour there, but never mind. This literally comes from the Greek saccharon, meaning sugar. So quite literally, the term monosaccharide means one sugar. So glucose is an example of this. And it's worth just pointing out at this point what a carbohydrate actually means. In terms of the atoms that are going to be present when we look at the structure. Carbo means from, so we expect carbon atoms to be in this structure, and hydrate, similar to hydration, means sort of water, so from water. And if you imagine water, as we all know, has this structure, H2O, that means we must have oxygens and hydrogens in there as well. So the 3 elements that make up carbohydrates are carbon, oxygen, and hydrogen. Quite literally what carbohydrate actually means. So let's look at the structure of glucose. So this is a carbohydrate. It is a monosaccharide, so it's a sort of simple sugar, and it contains carbon, hydrogen, and oxygen. And it looks like this. So just bear with me while I draw this. Try and speed up a little bit. 1, 2, 3, 4, 5, 6. So one more. So this is what glucose looks like in terms of a displayed formula. And hopefully you can appreciate that this links absolutely to the formula being C6, the molecular formula being C6H12O6. There are 6 carbons here, 12 hydrogens, and 6 oxygens. So there's a few things I'd like to point out about this this particular structure, which we'll sort of point out in a minute. But hopefully what you can appreciate initially is there are lots of oxygen atoms. Okay, lots of oxygen atoms here, and we know that oxygen from our water studies, if you have an OH bond, so R group here means just any sort of carbon chain. If you have an OH bond in your structure, the oxygen has 2 sets of lone pairs in p orbitals if you do chemistry, and of course the electronegativity value is very high. So the electrons in this oxygen-hydrogen bond here, this OH bond, are skewed towards oxygen. This makes oxygen delta negative. Don't forget you must put delta unless it is a formal charge. And hydrogen is therefore delta positive. That is worth bearing in mind. So the next few things I want to point out here is that there are 6 carbons, hopefully fairly obvious to you. We start to number the carbons so we know which carbon we're referring to later on. And we number from the group at the top here. And the chemists amongst you, I'm going to go slightly out of sync here compared to my objectives, but never mind, will hopefully recall that this functional group is an aldehyde. It's an aldehyde functional group, C double bond O, and we're connected to hydrogen as well. So the aldehyde functional group we can represent with an R group, meaning sort of everything below this line here. And that connects to our C double bond OH. That is the aldehyde functional group. So there's 2 facts here that I want you to sort of bear in mind. Number 1, we've said there are 6 carbon atoms in our structure. So that's number 1. And number 2 is that there is an aldehyde functional group. And these 2 things allow us to classify glucose, coming back to our first objective, in sort of two sort of families, I guess. The first is because there are 6 carbons, we refer firstly to any sugar, of which glucose is one. It has the suffix of OSE, O-S-E. And we can put a certain prefix in here depending on how many carbon atoms we have. So if we have 6 carbon atoms, we refer to this as a hexose sugar, or a hexose monosaccharide. There are many different hexoses. We'll come across another 2 in just a moment, but glucose is one of them. If you have 6 carbons and you are a sugar, that's quite literally what this means. Hex meaning 6, O's meaning we are a sugar. That means we are a 6 carbon sugar. The second point is that we have an aldehyde functional group at the top there. So not only are we a hexose, but we are also an aldehyde. And so therefore, that's similar naming system. Instead of putting hexose, we now say that we are an aldose, A-L-D-O-S-E. Again, splitting this up means ald comes from the term aldehyde, and ose means we are a sugar. So we're a sugar that contains an aldehyde functional group. So just zooming out a little bit there, hopefully that's kind of a fairly straightforward introduction. You do not need to know or learn how to reproduce the structure of linear glucose there. What you are expected to draw, I will make very clear when we look at the cyclic forms a bit later on. So I'm going to pop up to our objectives, and we can hopefully now tick off the first point here, which is that we have now classified carbohydrates into two groups. You can do it based on the number of carbons, and in the case of glucose, we're a hexose, so that's 6 carbon sugar, and we've also done it in terms of the functional group that's present. And in the case of glucose, we're on aldose because we have an aldehyde functional group. So hopefully that ticks off the first objective there. What I am now going to do is just mention a sugar now, which is ribose. I'll keep in the same order. I'm going to draw this slightly differently, and you'll see why in a minute. So objective number 2 is we now need to look at ribose. And hopefully you've already picked up on the suffix of this word, ose. So this is yet again another sugar. And it's a specific type that comes up when we look at DNA and nucleotides and things like that later in the course. And I'm not going to labour the point now because we'll have plenty of stuff on ribose when we do DNA and RNA. However, the general structure is as follows, and I'm going to do sort of skeletal formulae here. I'm not going to draw in a lot of the detail because I only just want to make the point that every bend in the road there's a carbon, unless we're told otherwise. So there's an oxygen at the top, and each of these points there is a carbon atom. So the reason why I mention ribose now is that there are 5 carbons in our structure. And there's obviously groups hanging off here, off the carbons, hydrogens, hydroxyl groups, but I'm not drawing them in for simplicity. The main point is we have 5 carbons, and we number from this carbon here. So carbon 1, 2, 3, 4, and 5. As opposed to glucose up here, which I'm now going to number, which is we number from the top here, 1, 2, 3, 4, carbon 5, carbon 6 in our glucose structure. So the only point I want to make about ribose here is that because there are 5 carbons, instead of calling it a hexose, hex meaning 6, we use the prefix pent. So this is a pentose sugar. Again, pent meaning 5, ose meaning a sugar. So quite literally, it means it's a sugar that's made of 5 carbons. I'm going to park ribose there, and I'm just going to put a nota bene here that we will come across this when we look at DNA and RNA structure. So we will go into a lot more detail in a subsequent topic. For now, just know that this is yet again another sugar, but in this case, it is a 5 carbon sugar rather than a 6 carbon sugar. That's all you need to sort of keep in mind for now, for this topic. So hopefully that was fairly quick. So ribose now ticked off. What I am now going to do is just give you 2 definitions for point number 3. We need to look at aldose and ketose sugars in a little bit more detail. We've actually already covered aldose sugars. We said that aldose sugars down here literally means a sugar, ose, with an aldehyde functional group. That's the ald bit at the start. And we looked at the composition of the aldehyde. And those of you that I teach, I set as prep very early on to learn your functional groups, and aldehyde was one of them. There is, however, another type of sugar that has a different functional group. So, point number 3. If we have an aldehyde functional group, which, as we've already said, looks like this, then we call this type of sugar, or the family, it's a family of sugars, there are many, an aldose sugar. And within this group, there are lots of different sugars, one of which is glucose, which we've come across. There are others as well, but I'm not going to mention them. Glucose is our case study, but it's just worth bearing in mind this is kind of like a surname for humans. And of course, within a family, you all share the same surname. You're all an aldose in this case, but you have your own different identities, right? There'll be glucose and there'll be essentially glucose has siblings here within the family. They're all aldose sugars. They all contain the aldehyde functional group, but they are different sugars. I mention this because you need to know one more, which is you can be a ketose sugar. And again, quite simply, this means you're O's, you're a sugar, and you have a ketone functional group. So I'm going to do that in pink here. You have a ketone functional group rather than an aldehyde. And the ketone functional group looks like this, where you have, essentially this is R group 1, R group 2, but you have a C double bond O group here, and connected to that carbon are other strings of hydrocarbon chains, essentially, with other bits poking off. I will tell you now that the example you need for this, and I'm not going to draw it out, we'll see it later, is fructose. Fructose is, unlike glucose, it's a ketose sugar. It's got a ketone functional group, does not have an aldehyde functional group. So this is absolutely a ketose sugar. But the sort of more astute of you will recall, it is actually an isomer of glucose anyway. So fructose actually has the same formulae, the same molecular formula as glucose. That's all I'm going to say on that for now. It will come back later on. So that's aldose and ketose sugars done. So now we're going to look at the isomers of glucose for point number 4. I'm actually going to switch pages here because I have pictures. Here we go. So these are the isomers of on the left-hand side here, and the formula is C6H12O6. The two isomers of glucose that you need to know are called galactose and fructose, which you came across briefly at GCSE when you looked at various disaccharides, which we will get to. Because they are isomers, these guys also share the same molecular formulae. They are both C6H12O6. And you can count them up if you want to. The main point of the news here is to firstly highlight our functional groups. So you can see on glucose here, carbon 1, we have our aldehyde functional group. So this is absolutely an aldose sugar. So I can highlight that here. It takes a while to highlight it. But that is also an aldehyde functional group. So this is also an aldose sugar. Fructose, on the other hand, is different. And hopefully you can appreciate by looking, I'm going to highlight it in a different color to show you. We have our ketone functional group here. Where these carbons here, essentially all of this is represented by an R group, and all of this downwards is represented as an R group. So this absolutely fits into our structure that I showed you earlier, which is that we have an R group connected to a carbon, and the carbon has a double bond to an oxygen. So this is absolutely, I'm just going to get rid of those for now, absolutely what we expect from a ketose sugar. And you need to know that fructose is a ketose sugar, galactose and glucose are both aldose sugars, but they are all isomers molecular formula. The same molecular formula, which if I just move to the middle just to make that clear, is C6H12O6. So they are all hexose sugars. They are hexoses. They all have 6 carbons in them. You do need to be aware of the type of isomer that they are. So I'm just going to move this out of the way, just so I can do this at the bottom of the screen. Hopefully, you can appreciate that the difference between glucose and galactose is on one of the carbons only, and it's just where the particular group in question lies. What I mean by that, I've explained that probably quite poorly, is if we zoom into this carbon here on both of these molecules, glucose and galactose, hopefully you can appreciate they are mirror images of each other. Essentially, the location, the side of the carbon to which the hydroxyl group and the hydrogen lie have been switched. So on the left-hand side for glucose here, the hydroxyl group is on the right, and for galactose, it's on the left. That is the only difference between the two. Therefore, kind of like how your left and right hands are mirror images of each other, we also have the kind of left and right side for the hydroxyl group. So this hopefully allows you to remember that speakers have left and right inputs, and therefore we term these two sugars stereoisomers. That's how I remember it. Okay, these are stereoisomers. Stereo, as in I remember that stereos have left and right inputs, so left and right inputs. Therefore, glucose and galactose on carbon number, don't forget 1, 2, 3, 4, 5, 6. On carbon number 4, we switch these around. So they are mirror images of each other. The difference between glucose and fructose is a bit more pronounced. And hopefully you can appreciate it is absolutely what gives rise to the ketone functional group, because the C double bond O here in glucose on carbon 1 has been moved to carbon 2 in fructose, right? If we number the carbons again, carbon 1, 2, 3, 4, 5, and 6, we are now, the C double bond O is now on carbon. I labour this point because we have therefore changed the structure. We have changed the three-dimensional arrangement of atoms. So therefore, we are not a stereoisomer anymore. It's not simply a mirror image. It is a complete rearrangement of the atoms within the molecule. We therefore term fructose, compared to glucose, a structural isomer. And if you were going to look at the cyclic, the sort of cyclic forms, we will look at them, well, like this. Quite why I've so far drawn them here as linear chains, and now I'm showing you cyclic forms, which is probably something that you're more familiar with. Again, glucose here having the typical hexagon structure, as does galactose. We will get to that in just a moment. In fact, we'll do that next. But the main point of the news here is because of the types of isomers that we have, right? Glucose and fructose are structural isomers of each other. The whole point here is, and it's hopefully now fairly obvious when we look at the cyclic form, the shapes look very different. This looks like a hexagon, whereas fructose looks more like a pentagon, with carbons essentially sticking out. So hopefully you can appreciate, and this will come back later when we look at Benedict's test, fructose actually has a completely different three-dimensional structure compared to glucose and galactose. It's a stereoisomer of both of these. So just bear that in mind. Whereas galactose, because we're only a mirror image on one of the carbons, actually has a very similar looking structure. Okay. The difference with galactose is, as we said, on carbon 4, that's where we have our difference. And if we number the carbons, this is carbon 1, this is carbon 2, carbon 3, carbon 4 is on the left-hand side here. So again, carbon 4 is this guy here. And hopefully you can appreciate that whether the hydroxyl group is on the bottom or the top indicates whether we are looking at either glucose or galactose. And just in terms of naming the carbons, we always go from right to left as you look at the oxygen. So the trick for this is to find the oxygen here as part of the hexagonal structure, and you start on the right-hand side of that. So you'll start with this carbon here. That is carbon 1, and you'll see why that's true in a minute. So you would do the same here. And of course, for fructose, here, we actually start over here. Oops. Anyway, you get the point. Okay. So that's all I wanted to say with regards to that. So I'm going to switch back over to my main notes here, and we can now tick off point number 4, which is the isomers of, oops, here we go. So now we're going to look at how we form cyclic glucose from linear glucose, which is an important point. So here I've drawn the structure of glucose again. And although it might look a bit different to you at the moment, this really is the same structure that you've already seen with regards to linear glucose when I drew it out as a complete straight chain. Because if we look at where our aldehyde functional group is, which is over here, C double bond O bonded to hydrogen and then bonded to an R group, right? This is the aldehyde group. And therefore, we number this carbon as carbon 1. And if we keep going, and don't forget this carbon at the top is carbon 6, this is what the structure looks like. Hopefully, you can appreciate that this is the same structure as we've already seen. All I've done is I've essentially looped around this to here. Okay, we've sort of taken the linear chain and we've sort of bent it round a little bit so that carbon 5 and the hydroxyl group that's connected to carbon 5 is in close proximity to carbon 1. So I'm just going to remove my ink markings here because we're going to do a lot of drawing and I don't want to get too confused. Okay, essentially, I've drawn this structure whereby what was a kind of linear chain like this has now bent around like this. Hopefully that's kind of what you can appreciate. That's what I've done. So essentially, I've sort of bent the structure around like this. I've kept carbon 1 in position, and the rest is just coiled around. Now, before we do this reaction, this is the one I'm going to focus on for our purposes. However, it's worth bearing in mind that this is a linear molecule. These carbon-carbon single bonds are they're sort of rotationally not restricted, so they can move around, they can rotate, and hopefully you can appreciate we have another scenario here where again our aldehyde functional group is here. This is carbon 1, 2, 3, 4, 5, and carbon 6 down here. I've just bent this round the other way. So in this case, we're going anti-clockwise rather than clockwise. Again, carbon 5 here is in close proximity to carbon 1. And these two, if I just zoom out now, are equally likely to occur. Okay. Favorable. Okay. And we would expect that 50% of the time the molecule is going to do this, and 50% of the time it's going to do this. So let's look at what happens. We're going to use this guy as an example where we're sort of rotating clockwise. Carbon 5 is really important. And I mentioned at the start, there's lots of oxygens in the structure of glucose. So don't forget this is glucose. And we're going to focus on, as I mentioned, carbon 5, and specifically the hydroxyl group that is attached. And the important point here is we have on this oxygen 2 lone pairs. So I'm going to try and draw them like this. This part of the video will take a little bit more time to explain, but hopefully it's useful to you. Just realized that turned it into some kind of a dot. It was weird. Anyway, 2 sets of lone pairs here. And we know from our lessons on water that this oxygen is going to be delta negative. This hydrogen is going to be delta positive. It's a horrible delta. Looking at carbon 1, we can say the same thing. We've got a double bond to an oxygen. This oxygen is going to be delta negative. This carbon, this central carbon, is therefore going to be delta positive. And hopefully remember from water, do go and look back, that these delta charges mean that the electron sort of density is skewed towards the oxygen. And that creates on carbon 1 a delta positive carbon, and on carbon 5 over here, a delta negative oxygen, where we have 2 lone pairs. So these guys are called lone pairs. And that means that they are essentially a pair of electrons not used in bonding. Okay, so if you want the sort of definition, a lone pair is a electron pair not used up in chemical bonding. And if you have a lone pair of electrons, these would quite like to form a bond. And the way that they're going to form a bond is if they are nearby to a delta positive atom. So I'm just going to highlight this delta positive carbon here because it's really important. Delta positive means the electrons are skewed. The electron density is skewed towards the oxygen here, which is electronegative. And I'm laboring the points here, but it's really important to get this clear in your heads. Because oxygen is electronegative, both electron pairs in that bond and that bond are very close to the oxygen. So carbon here, which is bad for the carbon, it wants to gain electrons. And I put wants here in sort of quotation marks. Atoms have no desire for anything, but essentially this carbon wants electrons. And it can gain electrons if there's a free pair of electrons that are available to form a chemical bond with it. And in this case, hopefully you realize there are. there are two sets of lone pairs over here on the oxygen that are available to form a covalent bond if there is an atom nearby that is deficient of electrons and wishes to gain more electron density. So I'm just going to pause a little bit there because that is sort of reasonably complicated chemistry at this stage. So I would advise you maybe just sort of watch that bit over again and just make sure you're completely happy. I'm going to get rid of my writing because I'm going to draw some various other things in. So I'm just going to get rid of this for now. I'm going to keep those electrons in. The rest of the video won't be quite as long as this, but I want to make the point here. So I'm going to redraw these two bonds just to make them a bit easier to see. And I'm going to reiterate the point that within these two bonds, I'm going to try and do them as different colors, I think. We've got a green pair of electrons here for this covalent bond. And we've got a yellow pair of electrons for this covalent bond, both near the oxygen. Don't forget the crosses in this case both belong to carbon. The dots, let's say, belong to oxygen. So it's still covalently bonded, but they are in a polar bond, right? It's a polar bond because of the electronegativity of oxygen. So what's going to happen to form a cyclic structure here? And by the way, the reason that glucose wants to do this is because it's far more stable as a cyclic molecule. So this does just happen. Is one of the lone pair of electrons in pink here. I'm being careful with my colors. In fact, I might change the color of this lone pair so I've done green, yellow to purple. So let's use the pink ones. The pink lone pair on the oxygen of carbon number 5 can attack the delta positive carbon here. So we draw an arrow from the lone pair of electrons to the delta positive carbon. And this is where I'm just going to mention briefly something about electron movement. If we draw a double-headed arrow, this means it shows of 2 electrons. If we draw in chemistry a single-headed arrow like that, this shows the movement of a single 1 electron. So because I've drawn a double-headed arrow up here, that means both of these electrons here in this lone pair, both of these are going to form a bond, a covalent bond, and attack or form a bond by attacking this delta positive carbon. If you are able to form a bond by donating a pair of electrons, we have a name for this. Okay, this oxygen here is therefore acting as a nucleophile. Don't worry too much. Get in your head the idea of what's happening here. Don't worry too much about the terminology, but it is called a nucleophile. Okay, so the fact that the oxygen here has a lone pair of electrons that it can donate to form a covalent bond means it is a nucleophile. Quite literally what this term means, nucleophile, if you're philic, you love something. And if you're a nucleophile, that means you love a nucleus. In other words, some kind of positive charge. And we have a delta positive here on the carbon. So therefore, the oxygen here with its lone pairs is a nucleophile. It's essentially attracted to a positively charged region nearby. And in this case, we've got a delta positive carbon. So hopefully that's clear. I am going slow deliberately just to make the point and hopefully make sure that this is clear in your heads because it is quite complicated chemistry. The next thing I'm going to point out is if the oxygen here is a nucleophile, the carbon here is loving electrons. It wants to it wants to gain more electron density. So the carbon here, aside, oops, I don't know what's happened here. The carbon here, aside from sort of being skewed of electrons because of the oxygen, it's electron deficient. It's lacking electrons. and it wants to gain more. Want in inverted commas. Therefore, we call it an electrophile. It's capable of receiving a pair of electrons to form a covalent bond because it is delta positive. So, those are the two terms I need you to get into your head. A nucleophile is nucleus loving, it's positively loving. So it's something like a pair of electrons that is able to be donated to an atom to form a covalent bond with it. And an electrophile is kind of the opposite. It's where you have some kind of either sort of positive or delta positive charge where you are electron deficient and therefore wish to receive electron donation from another atom nearby. Okay. So just to clear up space, I'm going to get rid of some this stuff just to make it easier because we're going to do a bit of drawing. I'm going to try and speed up slightly. So the first bit of drawing is that this arrow shows that the lone pair of electrons attacks this delta positive carbon, and it's going to form a bond to it. So I'm going to redraw this structure now down here as to what this now means. So if this now happens, we then get this product formed. So I'm going to draw my aldehyde group here on carbon 1, carbon 2, carbon 3, carbon 4, carbon 5, which links to CH2OH. I'm not drawing all the atoms in here and the hydrogens just for simplicity. I'll sort of move that arrow out of the way. There we go. So let's look at carbon 5. We've got the oxygen still attached. We've still got the purple lone pairs, but the pink lone pairs here have formed a covalent bond with this carbon. That's what it means. So I can now bond in here to this carbon. That is now a single covalent linkage. And the electrons are kind of existing here. Okay, I've drawn them as dots because don't forget they both came from oxygen. Right, there's not a dot and a cross because carbon didn't have an electron to give to this covalent bond. Okay, so it's worth bearing in mind now that because the pink bond here, the single bond, is made up of electrons that are originated, so the electrons came from the same atom. In other words, they are both from oxygen. It's a shared pair of electrons, but the electrons is not a dot and a cross. They are both originating from the oxygen. Therefore, we call this a dative bond or a dative covalent bond. Worth bearing in mind. So we've now formed this structure, and of course we still have the hydrogen here. Don't worry about this for now. We still have the hydrogen attached. Nothing has happened yet to this hydrogen. So that's what's happened in our first step. The delta positive carbon has been attacked by a nucleophile, by two electrons that are a lone pair on the oxygen attached to carbon 5, forming a covalent bond, or if you are really posh, a dative covalent bond between the oxygen and the carbon on, carbon number 1. So this looks like a cyclic structure. I just realized I didn't draw these in. They remain the same. Don't forget this is glucose, of course, so the OH group is at the bottom on carbon 4. If it was the other way around, hopefully you can appreciate that it would be galactose, but that's not important for now. Okay, the next thing that happens is we kind of have this intermediate that briefly forms, and the problem is this carbon. The problem is that carbon, if you remember back to lesson 1 where we did 1, 2, 3, 4 honk, carbon wants to form 4 bonds to have a full octet, a full outer shell of electrons, 8 electrons. It's kind of got more at the moment. Okay, so if we actually put the electrons in, we've got let's draw them in. We've got green and yellow for the oxygen double bond here. So I'll try and separate them a bit more so we can see. So in this double bond, I'll make the oxygen a bit bigger. We have green and yellow. So the yellow ones were here. Don't forget dot and a cross because it's a genuine sort of traditional covalent. Two sort of 4 electrons here that it's got. It's got 1 electron that it's sharing. I'll just do the rest in blue, that it's sharing with this hydrogen here, and it's got 1 electron that it's sharing with the carbon. So how many electrons does carbon have in total? Well, it has essentially 2 here. It's got 4 here, so that's 6. It's got 2 here, that's 8. It's now got 10 electrons in its outer shell. This is not stable. This will not exist for very long at all. It just sort of can't do it. The reason this has been able to form is because the electron density of oxygen has pulled electrons away from the carbon, allowing a region of positivity to be attacked by the lone pair on the oxygen of carbon 5. I'm going to get rid of these circles. So what has to happen is we now have to deal with this. We have to now remove electrons, a pair of electrons, and give them back to something. Now, hopefully you can appreciate what's going to happen. The obvious point here is that the double bond here is not required. If we break any other bond, right, then we're going to mess up the structure. So you don't need to know why this happens with the double bond oxygen, but it does. So I'm going to pick the green ones here. These green electrons in the sort of first single bond of the double bond of the C double bond O group get given back to oxygen. Oxygen takes them away, right? Don't forget, it's electronegative. It's quite easy for oxygen to do this now. So I'm going to draw an arrow from this double bond back to the oxygen, and it's double-headed because both of these electrons in green here are given back to the oxygen. What I'm now going to do is just edit. I'm not going to redraw the structure. I'm going to edit this diagram. So I'm going to break the carbon-oxygen single bond here that contained those green electrons, the dot and the cross, and I'm going to draw the dot and the cross back on the oxygen. So this bond, this covalent bond has broken. There we go. The electrons now exist back on the oxygen, and I'm going to draw a dot and a cross because they're now a lone pair. But don't forget the cross originated from this carbon originally. So they're going to be back on this oxygen, and because the oxygen has gained a single electron that it did not have before, it's gained the cross now, right? This electron here in the cross did exist on the carbon because it formed a covalent bond with the oxygen, but it now completely belongs to the oxygen. So oxygen here has gained an electron that it did not have before. Therefore, the oxygen has a negative charge. So I'm just going to get rid of some of my writing here. The oxygen has a negative, it's a formal negative charge now. So I'm going to draw this as a negative ion. So this is kind of what happens in the first step. I guess it's kind of 2 steps I've just shown, but we form a bond and we break a bond, essentially. So we form a bond between the oxygen and the carbon, oxygen on carbon 5, carbon on carbon 1. And we then break a bond. We break one of the 2 single bonds that were originally in the C double bond O group on carbon 1. And we form this structure. Now, the problem here is that the oxygen that we have now made negatively charged, it's gained an electron, is only forming 1 bond. And if we go back to our honk here, right, oxygen, if I try and get rid of that, oxygen wants to form 2, in inverted commas, to have a full octet. Okay, so oops, something's going weird with my surface. We need to form another bond with the oxygen. So what is the oxygen here going to do? And again, I'm not going to redraw this. Actually, yes, I will, I think. So here we go. I'm going to get rid of this. Right. What happens is this lone pair of electrons that are making this oxygen here negative, right? The lone pair here are going to they're going to pull a proton from solution. And hopefully you're aware, around all of this in our cells, we have water, where the oxygen, delta negative, and the hydrogen's delta positive. And I'm going to draw the electrons in blue. So we've got an electron pair here and an electron pair here. So what's going to happen? And actually, I'm going to draw the other electrons in here. So this will get messy if I draw the deltas in. So I'm going to get rid of the deltas for now. I'm going to draw oxygen's electrons that it already has. I'm just trying to color code this. Let's keep those in blue. I'm going to make this bond a different color just for ease. Here we go. It's those electrons there. And oxygen has its own electrons as well, don't forget, because it's got 8 in total. So it's got 2 lone pairs up at the top here. So that's the structure of water. Hopefully nothing new to you. But we're now going to form a bond, right? So this negatively charged oxygen on carbon 1, hopefully you can appreciate, because it's close to lots of water molecules, there's loads floating around, it can pull a proton off of a water molecule, a delta positive hydrogen. So these 2 electrons are going to form initially a sort of dative bond with that hydrogen. And the bond that's going to break is this bond here. These electrons, the dot and the cross here, are going to be given back to the oxygen here. So I'm now going to draw what we get as a result of this. So bear with me while I do this. So the oxygen is going to be in black. I'm going to try and keep everything color coded. And we've now formed a bond between the green electrons here on the oxygen and the blue hydrogen. So I'm going to put that as a green bond here to the blue hydrogen. And I'm going to put those electrons in, the dot and the cross. Okay, just to make it clear that it's a covalent bond. Obviously, it would be skewed towards the oxygen, right, because of the electronegativity of oxygen, but it doesn't matter for now. So that's what's happened here. We can now draw the rest of the molecule in. So we've got our hydrogen here, carbon, 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 hydroxyl group, hydrogen, carbon, carbon 6 with its CH2OH. Our oxygen here that has already formed the covalent bond with the carbon. So that is now what structure we have. That's the structure. We now need to deal with what's happened to the water up here. So just to recap, we've pulled a proton away and the electrons in that covalent bond have been given back to oxygen. Okay, so the water molecule now looks like this. So we've still got this hydrogen here, which I'm trying to color code it and keep everything the same. We've got our 2 lone pairs on the oxygen, which were there anyway in black. So I'll just draw them like this, 2 lone pairs. And the electrons here, the blue ones, the dot and the cross that were in the oxygen-hydrogen bond have been given back to the oxygen. Okay, so it gains the dot that it had anyway, from that covalent bond, shared pair of electrons, and it gains the cross, which originated from the hydrogen in that bond. Hopefully that makes sense. So what we've now formed, don't forget, oxygen, how many electrons has it gained that it didn't have before? Well, the answer is 1. So this structure here, if I draw the electrons in there in this black bond here, we've got a dot and a cross. Hopefully you can now appreciate it. It's gained an electron that it didn't have before. It's no longer sharing it in a covalent bond. So this is now a negatively charged ion. So the oxygen now has a negative charge on it. It's gained an electron that it did not have before. The only thing to point out is I've negated to draw in that hydrogen, which is still knocking around. And we're essentially nearly there now. So there's one more step that's now going to happen, which is we have to deal with this part of the molecule. This oxygen is forming 3 bonds, and that is bad, it's unstable. So let's think about the deltas again. I'll do it in black. This oxygen here is delta negative, making this hydrogen delta positive. And you can imagine, for simplicity's sake, that this hydroxide ion, which if I now just make it a bit more simplistic, it's simply OH minus, can move over here, and the oxygen here can pick up the proton from this oxygen within the glucose structure. So same arrow drawing here. The electron pair here that it's gained can attack the delta positive hydrogen, and this bond here can break off. So these 2 electrons are given back to this oxygen. So what this does is it now gives us our final structure. If I try and still keep it color-coded, but actually, I hope it's fairly obvious. I won't bother color-coding it just for this. Looks like this. And this is our final completed structure of cyclic glucose. So this is what happens to glucose in solution, this whole sort of reaction mechanism here, to turn it cyclic. So this is what we now refer to as cyclic glucose. And the reason that this forms is because it is very stable. This is far more stable than keeping it in its linear form. And there is an equilibrium constant to this, which is that it heavily favors the formation of cyclic glucose, such that it's around about 95% of glucose in your cells, in your body, exists as the cyclic form. The linear form, therefore, only exists about 5% of the time. And that's being sort of slightly generous. It's probably more like 98% and 2%. But point is, 95% of your glucose molecules exist like this. And this is why we draw at GCSE as glucose, rather, as a hexagon, rather than that straight linear form, for example, up here. So moving back to our sort of checklist, back to carbohydrates, here we go. We have now covered formation of cyclic glucose. What we're now going to talk about is point 6, which is alpha and beta glucose. So I'm going to tick those off now. And I'm going to actually go back to my drawing here. And we're going to look at this structure. Okay. And this structure shows, because we drew the glucose like this, and we used this diagram here, where we sort of curve round in a sort of clockwise direction here, you can appreciate the double bond oxygen is on the top of this carbon and the hydrogen's on the bottom. So when we look down here, that's been the same all the way through. And so the double bond O becomes your OH and the hydrogen remains at the bottom here. The name for this particular type of glucose is if your hydroxide group, your OH group, is on top, then this is beta glucose. This is a beta form of glucose. Okay, so we literally would write it in an exam like this. Which begs the question, what is alpha glucose? And the answer comes back to this, and this is why I made the point. Alpha glucose forms when we bend the molecule round anti-clockwise, and the oxygen here, the lone pairs on this oxygen, attack from the bottom of the structure attack the delta positive carbon here. Okay, and this means that the aldehyde functional group forms in the other direction. So I'll just swap this round. Essentially, all you need to know, the hydrogen flip around. So hydrogen here, OH group here. And if the OH group is on the bottom, that then is no longer beta glucose, but we now call it alpha glucose. And I remember this because beta is better. And if I were the hydroxyl group, I'd rather be on top of the structure. Okay, so beta is better, so the OH group here is on top. If you are alpha, the OH group is therefore on the bottom. The other way to sort of make this clear, which might be clearer in terms of a reaction mechanism, is that you hopefully from chemistry you know that single bonds can rotate. So this bond here can rotate. Oops, here we go. This bond here can rotate like this. So essentially, the O double bond O and the hydrogen here can be flipped around, okay, because this carbon will essentially invert, and that happens randomly. Again, 50% of the time, you expect to attack this lone pair on the oxygen. You expect to attack it when it's in this form. 50% of the time, I can get rid of this now. You anticipate that the double bond O is going to be like that. And don't forget, the double bond O is what's going to give rise. This is what's going to give rise to your hydroxyl group, OH. So beta is better and alpha is not. Finally, on that, I'm going to show you what you need to be able to draw. Okay, so all of this is all sort of fairly conceptual. What do you need to be able to draw? The answer is you need to be able to draw alpha and beta glucose in their cyclic form. And I do this because I remember it like an upside down face, smiley face. Except instead of drawing the mouth like this, we'll draw it in sort of more structural form. So we draw the first part underneath like that. It looks like a face. You know, if I was going to put two eyes here and a nose, it kind of looks like the mouth. And then going to do the same thing, starting down at the left-hand side here, upside down. So the same thing, upside down, except when I get to the top here, I'm going to put in my oxygen before I then drop back down. Okay. The only things left to do in terms of being able to sort of sketch this in an exam is to put your groups in. So we need to make clear that we have our 6 carbon sticking off the top, and every bend in the road, there's a carbon, don't forget. So at the top here, we need to write CH2OH. That is because if we didn't do that, the insinuation from a chemical point of view is that you've got a carbon here and you've only got hydrogens attached. So essentially it would be this. It would be a methyl group attached. But it isn't. It's CH2OH. So we need to write in CH2OH. We need to make sure it's glucose, so the hydroxyl group here needs to be on the bottom, hydrogen on the top. Again, if we didn't put the hydrogen in here, it would be assumed that it's a CH3, which it isn't. So we need to put the hydrogens in where there's only a hydrogen. And of course, let's just go for alpha glucose here. So hydroxyl group at the bottom, hydrogen at the top. And we need to put these guys in as well. So there's a hydroxyl group here and a hydroxyl group here, sticking out a different way. And again, we just assume there's a hydrogen attached. So that is essentially all you need to be able to do in terms of drawing this structure. Okay, I mean, if you want to be really keen on this, then obviously there's a hydrogen attached here, but don't worry too much. That's kind of the bare bones structure that would be expected of you in an exam. If they wanted more detail, they would have to give you a structure to look at, and you base your drawing off of that structure. So all you need to be able to do is identify this particular side of the molecule, which is the side closest to this oxygen, remember, because it starts on carbon 1. And you need to be able to flip them around, essentially. So in this case, if it's at the bottom, it's alpha glucose. If we flip them round and we draw the OH at the top, then this is beta glucose. And that's all you need to be able to do in an exam. So definitely practice this, please. It's easy marks to lose, but equally easy marks to gain just by learning how to draw that structure. Okay, so that is alpha and beta glucose done. So if I now head back to carbohydrates here, we've now done 0.6. Okay, so there's only 3 more points to go. And hopefully this will be relatively quick rather. Certainly 0.7 and 8 are quite straightforward. So I'll get back down to my notes here. So here we go. We've now jumped to point number 7, which is disaccharides. I'm just going to check I'm correct. I'm sure it was. Makes sense. Yep. So remember, saccharides mean sugar. Monosaccharides were glucose, fructose, and galactose. We've looked at those. A disaccharide, therefore, contains 2 sugar molecules put together, 2 monosaccharides, 2 times monosaccharides. So, easiest example is you need to know, firstly, glucose added to glucose makes the disaccharide maltose. And I'll show you the structures in a minute. Okay, this is GCSE biological molecules. Glucose remains the same, but this time if we add galactose, we make lactose. And the reason that, well, how I remember this is literally the word lactose in this monosaccharide, galactose, it's literally in the word, galactose. Therefore, we make lactose. The third one you need to know is that if we take glucose again, and instead of adding lactose or glucose, we add, I'm going to do it in a different color here, we actually add fructose, we make the disaccharide sucrose, which, if you're interested, is table sugar that you'd find on the table. You need to know these 3. But hopefully what you appreciate is that all of these monosaccharides, the glucose is here in black, and the galactose and the fructose, these are all the isomers that we saw earlier. They all are C6H12O6 in terms of their molecular formula. And remember, of course, that fructose, and this comes really important when we look at the tests, fructose is the structural isomer. Okay, it has the pentagon shape rather than the hexagon shape, don't forget. Okay, the other 2, glucose and galactose, absolutely have the hexagon structure that we've seen up until now. So, how do we make these? And the answer is as follows. If you imagine 2 molecules, this is quite common in biology, right? If you have 2 molecules, let's do a green one and a black one, that have hydroxyl groups on them and they get close to each other, there are enzymes that can catalyze a reaction between them. And the enzymes essentially remove water. They remove an oxygen and 2 hydrogens. So these will pop off here and produce water. So I'm going to draw it as we would see in the actual thing, where one of the hydrogens there in green is from the other molecule. Okay, so this is H2O. This is water. And what that means is we've now been able to form a bond between the black molecule here essentially here, to the green oxygen, which was part of the other molecule, the other monomer, making kind of essentially two molecules now forming one. Okay, and you can imagine that these are both sugars. Let's imagine these are both glucose, because we have lots of oxygens, lots of hydrogens. And now, because they're connected together, whereas these were both two monosaccharides, this guy here, all in one, is now a disaccharide. So this whole thing is now a disaccharide. That is essentially all you need to know and be concerned about. The mechanism is quite similar to how glucose forms the cyclic form. You have a nucleophile that is a lone pair on the oxygen, can attack the carbon that connects to the other hydroxide group, but don't worry too much about that for now. You won't be asked to recall the mechanism for it. The key point is we have liberated water. Okay, we have produced water in this reaction. And there's a definition for this type of reaction which states that if we take two individual molecules and we join them together to form a single larger molecule, with the liberation of a small molecule, we call this a condensation reaction. Okay, it's a condensation reaction. Very important. Okay. And how you can remember this, and I make this point very clear to you, this is how you can remember that a condensation reaction is what it's called. It's not universally true. That the small molecule that we liberate, sort of 90% of the time, and I'm kind of making that figure up, but basically a lot of the time, it's water. Because in biological systems, we have lots of hydroxide groups, as you've seen here. And condensation happens when we have water forming, when you get out the shower, or, it's a cold day, or, something like that, and you look at the window, you've got water droplets on there. Condensation, water has been produced. But I make the point, certainly for my classes, please, that this is a general rule. There are exceptions. Okay, you can still have a condensation reaction where you don't liberate water. You liberate something else, another small molecule, whatever that may be. And when we do nucleotides and DNA replication and transcription, you will hopefully see that that is a major example that we'll come across where water is not produced. So the only thing you need to do here now is be able to draw this, but you can hopefully do this because you need to be able to draw, for example, two glucose molecules coming together. So let's draw one in black here. I'm going to sort of do this fairly quickly. Let's do one in, that is horrible. One in green, like this. Don't forget the 6th carbon, never forget that spike at the top. And we should really draw our CH2OH in here. And the rest of our molecules. Let's just imagine these are both alpha glucose. Okay, so we're going to put the OH group at the bottom. So OH here, OH here, hydrogen here. We've got our OH group here, hydrogen here. I'm not going to draw the rest in because it's not totally important for what we're trying to do, but nevertheless, alpha glucose, so OH at the bottom, like that. So we're going to use my sort of schematic here of two blocks here with OH groups, and we're just going to convert that to our sugars. So imagine the sugars are the blocks, right? We've got our hydroxyl groups at the bottom. These are going to react here to produce water. And we are going to form a linkage between our black monosaccharide here to the green oxygen, which then links to this sugar here. Sloppy chemistry there, but my water's in the way. That bond should connect to the carbon. There we go. So we form this. And this is our disaccharide, right? Don't forget water's been liberated. And we give this particular bond here a name. It is called a glycosidic bond. Okay, it is the bond formed between two monosaccharides. But we can be a bit more specific than that. And this is what I'd like you to do, please, certainly for those that I teach. Don't forget we number the carbons. Okay, this is carbon 1, 2, 3, 4, 5, and 6. Okay, I'm going to do the same here. This is carbon 1, 2, 3, 4, 5, and 6. So not only is it a glycosidic bond, but we can name which carbons we're forming the bonds to, or between. And in this case, it's between carbon 1 on the left-hand molecule here and carbon 4 on the right. Therefore, we can call this not just a glycosidic bond, but a 1,4, and we put a comma between the two numbers. 1,4 glycosidic bond. That tells you which carbons we're actually connecting. And the reason I make that point is because this actually does change, right? We'll come across examples where we have something like a 1,6 glycosidic bond, for example. But in this case, it's a 1,4. And if you want to be really posh, you can denote which types of glucose, which isomers, alpha or beta, we are joining together. In this case, both of these were alphas, so we simply call this an alpha 1,4 glycosidic bond. If they were both betas, then we would call this a beta 1,4. If we had an alpha and a beta, then you would do alpha beta 1,4 glycosidic bond. But you hopefully get the point. As it happens, these were both alphas, so it's an alpha 1,4 glycosidic bond. So that is the definition of a condensation reaction, how a condensation reaction happens between two monosaccharides, in this case, two alpha glucose molecules. The same principle applies for glucose and galactose and glucose and fructose, or if it was beta glucose, for example. Same principle occurs essentially, and we form a glycosidic bond between the two, and we liberate a water molecule here. So let's scroll to the top. That is that point done. So we've now covered disaccharides. We've touched on the three you need to know, which is maltose, sucrose, and lactose, and the monomers that they're formed from. And we've now covered a condensation reaction, how we explain what that is, the definition. The water most of the time is released by joining two smaller molecules together to form a large molecule. And we form a glycosidic bond when we do that with sugars. Okay. The next thing I'm going to touch on very briefly is the molecular formulae. So this is point number 8. So molecular formulae. And this is going to be fairly quick. You are expected to be able to give the molecular formulae for disaccharides, trisaccharides, tetrasaccharides, yada, yada, yada. And this may seem initially complicated, but they will have to give you the structure of the monomers. Okay, so just watch this way. Let's imagine we have two glucose molecules that we're going to put together to make maltose. Okay, so we're going to join them together like this via our glycosidic bond. You could get asked, if these are both glucose molecules, what is the formula of the disaccharide, the molecular formula? And the Dr. Hayes method of doing this is as follows. So I'm going to call this the Doc H method. Number one, work out your formulae for the monomers. Step two, add these together. Step three, remove the water. And that is absolutely the one that people forget, is to remove water. Don't forget, if you form a glycosidic bond because you're linking two monosaccharides together, you will liberate a water molecule. Yeah? So, let's use the method. Formula of the monomers, well, they're both glucose, so C6H12O6. We then need to add them together. We have two of them because we're adding two monomers together. So this becomes C12H24O12. Okay, we've just times this by 2. But we've not accounted for the loss of water. And the key thing here is to remember how many waters we lose when we add two monosaccharides together. If we form one glycosidic bond, we remove one water. So if you add two monos together, we lose one water molecule. If we were going to add three monomers together, we would have to form two glycosidic bonds, so we would remove two water molecules. So the sort of equation way of doing this, I teach my lot, is it's n minus 1. N being the number of monos, the number of units you're sticking together, number of monosaccharides that you're adding together. So in our case, we're adding 2 together. So n minus 1 is 1. So 2 minus 1 is 1. So therefore, we will only lose one water molecule. So all you do is now remove one water, H2O, right? 2 hydrogens and an oxygen from this. So carbon stays the same, happy with that. 2 hydrogens, this becomes 22, and 1 oxygen, this becomes 11. So your molecular formula for 2 glucose molecules to put together, so maltose, would be C12H22O11. Hopefully that makes sense. They would have to give you the information as to what the monomers are, unless they give you glucose, galactose, fructose, because you need to know they are isomers. So they all have the same molecular formulae anyway. Okay, that's that done. Told you that would be quick. The final thing for this lesson is to look at polysaccharides. And I'm going to do this through pictures only. So I'm not going to draw too much for this because it's just a lot of stuff you need to know. So polysaccharides, here we go. I'll show you the disaccharides later. In fact, I'll do that now just while we're mentioning it. Here we go. You don't need to know these structures, but it's worth bearing in mind. This is what sucrose looks like. This is what lactose looks like. This is what maltose looks like. And again, we'll come back to this. Sucrose is made up of glucose and fructose. This is your fructose group. It messes up the structure. It's a pentagon, not a hexagon. Okay, sucrose has a completely different structure to lactose and maltose, which look fairly similar. Okay. Don't forget, of course, though, that lactose is made up of glucose and galactose. So the fact that the OH group is at a different position on galactose. Okay, it's not at the bottom here, it's at the top. That's how you recognize galactose. We'll give it a different property, which we'll see later. Maltose, it's just 2 glucose molecules put together, specifically 2 alpha glucose molecules. Okay, lactose, we have, glucose and galactose, as I said before. But they all have glycosidic bonds, and you don't need to learn this, but it's worth bearing in mind. Lactose and maltose have typical 1,4 glycosidic bonds, and if you look carefully here, sucrose actually has a 1,2 glycosidic bond, but don't worry too much about that. Okay, that's just food for information, really. You'll never be sort of asked the nitty-gritty on that. So we're going to look at polysaccharides now, and I'm going to go reasonably quickly through this because it's just stuff you need to sort of learn, really. The first polysaccharide, poly, by the way, meaning many. So poly literally means many. And we typically refer to this, so it's not a disaccharide, it's not two, it's not a trisaccharide, three, it's many, poly. Typically, we think of this as sort of 30 plus individual units, some 20 to 30, but usually we're referring to hundreds, if not thousands, of individual monomers stuck together by glycosidic bonds. This molecule that we're looking at here, which the sort of cartoon is here, this is glycogen. Okay, you hopefully know from GCSE, this is the energy storage molecule in animals, in animal cells, as opposed to in plants where we use starch. And the key point is it's made up of lots of alpha glucose molecules, just stuck together, lots of them. So it's not maltose where we have two, it's many, right? It's glycogen, lots of them. These are all alphas. So zooming in down here, we have an alpha 1, that's carbon 1, that's carbon 4. We have an alpha 1, 4 glycosidic bond. That means that this is a linear chain, right? We just, instead of putting two together, we've put loads together, right? We just keep adding into a linear chain. And you can see that there. But in glycogen, what we can do is there are enzymes that allow this to happen, where we can actually not only link between the 1 and the 4 carbons, we can link between the 1 here, carbon 1, and if we count the carbons round in green here, that's carbon 1, 2, 3, 4, 5, 6. Okay, this carbon here is carbon 6. Therefore, this glycosidic bond, it's not an alpha 1, 4, it's an alpha 1, 6 glycosidic bond. So not only can we stick glucose molecules together in a linear chain by using 1,4 linkages, 1,4 glycosidic bonds, we can stick them together and we can branch off other molecules by utilizing the 6th carbon, right? So forming alpha 1, 6 glycosidic bonds. So we form both. Okay, so make sure that that's clear. We have alpha 1, 4 and alpha 1, 6 glycosidic bonds. Very important. What this allows is the properties of glycogen, which you can see here and schematically in this cartoon. So the properties that make it good for energy storage are as follows. It's firstly quite compact as a molecule. You can see that here, right? It's quite, you've got a lot of glucose in a small space. So compact means lots of glucose in a small space. That's useful for cells, animal cells, because your cells are very small. So you need lots of glucose in a small space. Your efficient storage, essentially, so compact. The other thing, which is clear by the diagram, is it's branched. It's got a branch structure. That word is very important, branched. And it is branched because not only do we have alpha 1, 4, but we can also have alpha 1, 6 glycosidic bonds. Okay? We can have alpha 1, 6, which means we can branch, as we've shown here. Okay? What that means is you can have lots of free ends, okay, which you can see here, right? We've got an end here, 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 all the way along the molecule here wherever we've got branching going on, right? Lots of free ends, as opposed to if we just had linear chains, we couldn't have as many ends. This is important because your enzymes that can digest and break down glycogen can attach to all these ends. Okay, enzymes have a therefore high surface area for which to act on. Okay, and the enzymes, you don't need to know what they are. Glycogen phosphorylase is one of them, but don't worry. Can bind to each end, right? You can have an enzyme here that's going to start chewing off. It's going to break these alpha 1,4 glycosidic bonds. Okay, so the enzymes have a large surface area to act on. Therefore, we have efficient and quick hydrolysis, which we'll get to later. Hydrolysis is a posh word for break. It's the opposite of condensation, right? Breaking, essentially, of your 1,4 glycosidic bonds. This can happen simultaneously. Sorry, handwriting is getting a bit dodgy now. Happens simultaneously. What I mean is these enzymes can bind to all of these free ends at the same time. And so we can start hydrolyzing all of these bonds, releasing lots of glucose quickly. Okay, so this can happen simultaneously, and we therefore release lots of glucose, the monomers. And the reason this is important is because glucose is required. This is always good for the exam. Why is it needed? Because this is used for respiration. Okay, it's a substrate for respiration. In upper sixth, substrate for glycolysis. Okay. Therefore, from respiration, we can liberate lots of ATP, right? The energy, the sort of free energy stored within this molecule, the ATP molecule. And this can be used for things that an animal or a human like you needs, right? You need to move, right? Muscle contraction. You need to excrete, so excretion, right? Coordination and response, right? Your brain needs a lot of ATP, right? Loads of things. Essentially, this is Mrs. Gren. C. Don't forget C stands for coordination or control. But that's kind of like a good 3-4 mark answer there as to why glycogen is branched. What's the important property of glycogen being branched? The next thing to point out about glycogen is it's insoluble. This is important, and if you look back at your water potential notes, I'm not going to cover water potential here and now. If it's insoluble, it does not dissolve in solution. It therefore does not affect water potential given this symbol here. Okay, so this is water potential. Okay, what that means is in your cell, you can store lots of glycogen. If we have a cell here and we're storing lots of glycogen as this branched molecule here like this, it's not going to lower the water potential, right? If it dissolved, then it would lower water potential. We'd expect the water potential to decrease. The problem there is because the water potential outside of cells is very high, water would move in by osmosis, eventually leading to osmotic lysis, bursting of the cell. But this doesn't happen because glycogen is insoluble. It remains in kind of globules, essentially, specifically in the liver, in muscle as well, but predominantly stored in the liver. So they're the properties of glycogen that you really need to sort of worry about. It's compact, so we can store lots of glucose in a small space. What that means is it kind of links back to this point, if you needed to explain it. It links back to, therefore, that we can get lots of glucose in the cell for lots of respiration, lots of ATP production, etc. So that is glycogen done fairly quickly. Just checking my laptop is actually charged, or am I going to lose it? I think we're okay. So we'll continue. Not long left on this. Okay, that's glycogen. We're now going to look at the plant equivalent, which is starch. Okay, so this is the plant equivalent of glycogen. Okay, so it's the main energy storage molecule. It's the glucose storage molecule in plants. And it's actually made up of two types, two separate molecules, right? We've got something called amylose and amylopectin. And you don't need to know much about it. Don't worry about the reducing ends for now. I remember this though. Amylose is a simple word. Okay, it's a short word. Therefore, this is quite simply alpha-1,4 glycosidic bonds. Okay, it's a straight chain. It's a linear chain, right? Looks sort of like this. That looks skeletal. Looks like this. Okay. So that's what it looks like. It's kind of a straight chain. For now, amylopectin is more complicated. It's a longer word and complicated. So it's complex. Therefore, this is structurally similar to glycogen. It is branched. And it is branched because we can form 1,6 glycosidic bonds. Okay, so I'm not going to go through the reasons again. Essentially, why this is branched, it's useful. It's useful to the plant because of the same reasons as glycogen, right? Lots of free ends for lots of enzymes to bind to hydrolyze the glycosidic bonds, the 1,4 glycosidic bonds, and then release glucose for respiration, for ATP, and of course you need ATP for lots of things in a plant, right? So transport and active transport of minerals and ions into root hair cells and that kind of thing. So the only point here to make, amylopectin is very branched. Amylose, because it is simply a linear chain, right? It's simply, I'm not going to draw hexagons, it's simply a linear chain like this. Because it's linear, and because all of these glucose molecules, and this is a complete sketch, don't take this as any kind of accurate drawing, but because glucose, don't forget, has lots of OH groups, like this, these can interact with each other and form hydrogen bonds. Look back at the water lesson if you need a recap on hydrogen bonding. So actually, amylose can start to look like this. It can start to coil up like this. So you've got alpha-1,4 bonds, glycosidic bonds, but you've got hydrogen bonds occurring like this, intermolecular forces, hydrogen bonds between delta negative oxygens and delta positive hydrogens that form a coiled structure. So that's really important. It forms a coiled structure, amylose. And this means we are compact. Okay, very compact as a structure. Now, why plant cells have a mixture of these two things, which you can see here. I think this is quite a nice image. It shows that you have your red amylopectin, which is very branched. As you can see, it's making lots of branch points. But your amylose in blue there at the top is just a single sort of coiled structure. Why plants have this, why they have both amylose and amylopectin, you will never be asked, or well, never say never, but you shouldn't be asked. But the reason is because we need a trade-off in plants. The trade-off is between storing lots of glucose and being compact, more compact than glycogen actually, but you need lots of ends, free ends, because we want quick release, efficient release, hydrolysis, to get lots of glucose. So we've got a trade-off. Apologies for my handwriting, it's getting worse. We've been going for an hour and a half. It's a trade-off between these two things, right? If you are compact, this is what is achieved by coiling in amylose. So that's what amylose helps with. Amylopectin allows you lots of free ends, branching, which allows for quick and efficient simultaneous binding of enzymes to cause simultaneous hydrolysis of alpha-1,4 glycosidic bonds to release glucose quickly into the into the plant cell. So that's kind of why plants do it. And actually, as you can see here, amylopectin exists sort of around about 80% of starch is amylopectin, and around about 20% is amylose. So that's the trade-off there, 20-80 for that. Worth thinking about though, quite interesting. Right, the final type is cellulose, as you can see here. Okay, now up until now we've been talking about alpha glucose. So for both starch and glycogen, alpha. Here we're actually referring to beta glucose. So this is the different one, cellulose. We now are using beta glucose. Very important. What beta glucose means, and the fact that we can use beta glucose here, and why it's used, in fact, is because you can see here every other molecule rotates 180 degrees with respect to the previous one. Okay, because that's also shown by the oxygens here being on the other side. Okay, we rotate 180 degrees. Each individual molecule is flipped 180 degrees. Okay. So that's important. What that essentially allows for is cellulose is a linear chain of beta glucose molecules connected by beta 1,4 glycosidic bonds. But the difference is, because we're beta, each sort of molecule essentially has the CH2OH group flipped around 180 degrees. It's the reason we can do that is because we have beta glucose, but don't worry, it just happens. Just go with me on it. That's all you need to know. The importance of this is we can have thousands more hydrogen bonds that form. So no, it doesn't actually coil like in amylose. What happens is we have a linear chain. It can't coil very easily. What it can do is form intermolecular, between molecules, intermolecular hydrogen bonds. In other words, we've got one sort of cellulose fiber here, we've got another one here, another one here. They can sit on top of each other and hydrogen bond between the layers. And you can have thousands of these because of the 180 degree flipping of beta glucose molecules, every other one. And from the lessons, hopefully you appreciate, remember from hydrogen bonding, few hydrogen bonds are very weak, but collectively they're very strong indeed. So it is used for structural support in the cell wall of plant cells. Okay, it is a structural carbohydrate. It's not used for energy generation, it's used for structure. So that's just showing here, essentially this thing here is kind of one of these. They can stack on top of each other. So here we have cellulose kind of sort of molecules sticking together, and this is kind of what you need to know. We have 1,4 glycosidic bonds, beta 1,4 glycosidic bonds between the molecules, and the chains stack on top of each other with lots of hydrogen bonds between them. Collectively, these together, we have lots of stacks stacked on top of each other, lots of hydrogen bonds, very hard to break, so structurally very, very sound. The reason why each alternative beta glucose molecule is flipped is to maximize the hydrogen bonds, the number that can form. These collectively form one of these. And if I zoom in here, all of these guys, and there'll be more than just 3 layers, form one of these sort of circles here. I'll highlight it in a different color. One of these, that's even worse. I'll do it in blue. One of these, there we go. And lots of these come together to form what we call a microfibril. Micro meaning small, fibril made up of lots of chains. And the microfibrils stack together even more to form a macrofibril, which you can see here. So lots of micros form a macrofibril. The macrofibrils form together to form a fiber. Okay, and then lots of fibers. So I'll try and do it in pink here. Lots of the macros form fibers. Oops, that's not working very well, is it? Lots of fibers. Why can't I draw on that? That's something being really weird. Oh well, lots of fibers that make cellulose. So lots, very strong. It's in the cell wall of your plant. So how is cellulose adapted? Well, it's made up of beta glucose. How is it adapted to its function? Beta glucose. Every other molecule is inverted 180 degrees. This allows for thousands, lots more hydrogen bonds between layers or chains. This makes it very strong. Lots of hydrogen bonds are very strong. The chains pack together to form microfibrils, which further pack together to form macrofibrils, which eventually form a fiber of cellulose, which is in the plant cell wall. So this is important because it all gives structural rigidity, strength, very, very strong molecule, okay? And the reason this is important is because of osmotic pressure. I'm going a little bit quick because my surface is about to die. Okay, in our plant cell, right, we've got lots of water pushing against the side of the plant cells, right? We need to resist that change, otherwise osmosis would cause problems, right? The cells would die. So we need to resist osmotic pressure that's building up in the plant, in the xylem. For example, stems need to be structurally sound because they need to essentially stand up, right? They need to go essentially from the soil to the leaves, right? And also plants need to resist things like wind, gravity to an extent. Yeah. So, the wind that sort of blows them around, if they weren't structurally sound, their cell walls didn't contain cellulose, and all of this was true, they'd sort of flop around and then break very easily. Okay, that's the end of carbohydrates nearly, apart from just looking at testing for carbohydrates, which we will cover next time.